This is going to be a summary of non-cyclic photophosphorylation. We're going to begin with the sun because everything in living things begins with the sun, particularly when we're talking about photosynthesis. The sunlight is going to hit the leaf. It will go into the chloroplast and into the organelles inside the chloroplast called the thylakoid. Everything that we're going to talk about in non-cyclic photophosphorylation is going to happen in the thylakoid. The thylakoid has an outer membrane and an inner membrane. Now the way I've drawn it here, it makes it look like there are three membranes. But I want you to remember that the membrane has an inside and an outside phospholipid layer. So this is my inner membrane, and the electron transport chain is going to happen there. This part over here, this is the inside of the thylakoid called the lumen. So we're going to be working here and here when we're talking about photophosphorylation. So our sunlight is going to come in. It's going to hit a water molecule. The hydrogens are going to split off and go out here into the lumen. The oxygen is going to come down here to the bottom of the electron transport chain that I haven't drawn yet. And the electron is going to go to the reaction center of photosystem 2 called P680. P680 is a chlorophyll pigment, that's where the P comes from, that absorbs energy wavelengths at 680 nanometers. Now this P680 is going to gather the electron. It's going to gather other sunlight energies coming in from other pigments that are around it. Those things are called antenna pigments, until they've got enough energy to pop that electron into the electron transport chain. That electron has too much energy for the cell to use, so it's going to pop down the chain and react with the molecules that are there. When it does, it's going to lose some of its energy because it takes energy, remember that activation energy we've talked about, to make reactions happen, and that energy will be lost as waste energy. And that will slow our electron down, and at the same time a hydrogen will be pushed out of that chemical reaction, and it will also go over here to the lumen. Each time the electron moves down the chain, that reaction happens. Energy goes into activation energy, energy goes into waste heat, and another hydrogen pops out. Each time this happens, our electron slows down, and the concentration gradient goes up. Okay? When we get all the way down here to the bottom, where oxygen is waiting, that last electron will be involved in a final electron exchange, if you will. A new electron will come out, and it will enter the photosystem 1. The oxygen will float away as waste. Now we're down here in photosystem 1, and just like photosystem 2, the electron will go to a chlorophyll pigment called P700. It will get energized, and it will go into a second electron transport chain. Each time it goes down the chain, it will lose energy, just like it did in photosystem 2, and a hydrogen will come off, just like in photosystem 2. The difference is that these hydrogens do not go to the lumen, they just sort of hang around the electron transport chain. At the bottom of photosystem 1 is a molecule called NADP. Now, before we go any farther, I want to go back up here and talk about these hydrogens. We've got the hydrogen sitting here in the lumen, slowly increasing in concentration. Eventually, the concentration will become too much, and they will want to flow out of the lumen, back through the other side of the inner membrane. Okay, there's my phosphates. They're going to flow through the membrane through the transmembrane protein called the enzyme ATP synthase. So what we have here is we have a transmembrane protein that is specialized because it's an enzyme. Inside this enzyme, you have a whole bunch of ADP and phosphate molecules just hanging around, waiting to be rebuilt into an ATP. So you're going to take that hydrogen, it's going to come out of here, it's going to float through the synthase, and as it floats through, it's going to glue that ADP to a phosphate to create ATP. Think of it back when we did the enzyme chapter. There's my enzyme. Okay, there's my active site right here. ADP is a substrate and phosphate is a substrate. Hydrogen acts like a cofactor and glues them together. So between the enzyme and the hydrogen, we go from ADP and a P, and we create ATP. And that ATP then will be transported to somewhere else in the cell by way of Golgi bodies, those vesicles we talked about in chapter 4. We go back down to the bottom of photosystem 1. At the very bottom of photosystem 1, we have something similar happening. The last electron at the very bottom and the last hydrogen that it made will attach to NADP. So NADP plus the electron, which will act as the glue, will add that hydrogen to the end, and we will end up with an NADPH molecule. That molecule then, just like ATP, will float away by way of the vesicle until it's needed. This one is even better than ATP because when you break it down, you get 3 ATP molecules. When you break it down, and when you break ATP down, the Golgi bodies will do their job in reverse. They'll bring vesicles filled with all of these pieces parts back to the electron transport chain area, back to that synthase area, and deposit them so that they can be rebuilt the next time the electrons run down the transport chain. And that is the end of non-cyclic photophosphorylation.